Hi, this is Mike Dolinski here. It's springtime here in the prairies, and farmers have got crop in the ground, got seed in the ground. Some is germinating, maybe a little bit's up. Some folks in the cooler parts of the prairies aren't seeding yet. So we're all over the place in terms of progress this year. It started out warm, got cold, and the nights are still pretty cool in some parts. So this is the beginning of a series that Taurus is going to put on, and I'm going to put on, talking about foliar nutrition for the rest of the summer. And we're going to try and get them ahead of schedule so that you have a couple of weeks to think about what your next step may be from a foliar nutrition perspective. So we're going to pump these out on a regular basis, talking about things like VPR, flour, grain fill, pod fill, and all those nutrients, so that as the summer goes on and we see what stresses are taking place and what the moisture conditions are like, you can make some decisions about which products may fit your particular farming situation and your nutrient supply situation. So this is the first. I'm going to be doing a lot more intense stuff up front in terms of the plants and the soil situation across the prairies and some general stuff, and then talk about active build and the nutrients in it, and a little bit of detail about application of this particular product and some of the traits of the product. So in the future, the upfront part will not be included probably, except when we get to flower, because that's a sort of different reproductive or physiological stage in the plant. So they'll be a lot shorter. So this one will run probably half an hour to 40 minutes and give you some background to get you thinking about nutrition and what's going on your farm or across the prairies. So let's get started. And I'm just going to show you this because I want to point out what nutrients are an active build. And on top of the nutrients, we have a patented bioactive molecule that's incorporated in the product to help the plant in terms of up and down regulating its genes and helping deal with transpiration and movement in nutrients. It's patented and privately held by Active Agro. So these are the nutrients in it, a little bit of nitrogen, 2%, and all these are readily absorbable. Most of the micros are in the sulfate form. Boron is in the borate form because that's generally the form it comes in. So they're all readily absorbable through the leaf, through the cuticle, and probably through the stomata, depending on when you're spraying and the kind of foliar application you're getting and the misting you're getting on the plant. But there are mechanisms for the plant to take this in, and then once it's in, it distributes it. So we have nitrogen, phosphorus, a lot of phosphorus because the plant is always needing phosphorus and it gets tied up quite readily in the soil after about 25, 30 days. Potash is not very mobile, so we want some in there all the time. Boron, manganese, important in a lot of enzymes, so is zinc, and then molybdenum. So these are what in the product, and as I go through the presentation, you'll see that these have specific roles. But let's take a look at what the situation is here on the prairies. This is a summary of A&L's 2020 samples, and you can see in the soil analysis that phosphorus is still a problem. Sulfur is short in a lot of samples, and the variability is tremendous across the landscape. So soil sampling is pretty important there, and many people are now just putting down sulfur. We don't put any sulfur into build. Farmers should be supplying that all season long, and probably lots of it, especially with canola, but anything that's got higher protein will need more sulfur. Boron levels seem to be around and increasing, and so is zinc. And in my active prime presentation I did a little while ago, I talked about the wheat seed analysis we did in combination with 2020 Seed Labs, 100 samples from Alberta, a couple from Saskatchewan. 100% of those showed low levels of boron, and 51% showed low zinc. So here across the prairies, it looks like we've got zinc at low levels in our soils. In the tissues, boron consistently showing up as low, and I think there are some good reasons for that. We put some boron in the active build, but it's not enough to sustain you. You need additional boron either in the soil in organic matter, or some granular application, or high specific boron. So I should probably point out at this time that The way I look at I'll just go back. The way I look at foliar nutrition is as follows. Plants are made to take up nutrients through the roots, and before man was here, there was nobody feeding it except the atmosphere, you know, things like sulfate, sulfur dioxide, and sulfur from volcanic activity and so on. So when we put on a foliar, you can see it's not tremendous amounts of nutrients, although they're very efficient when you put them on as a foliar because they don't interact with soil and get tied up with other minerals and organic matter and so on. But it's not a lot. And the way I look at it is it gives the plant a little shot of energy. It's like getting a vitamin or something to carry you through. It'll last for maybe a week to 10 days, grow enough roots, produce some more sugar to get roots going, maybe move some of that down into the rooting system, move some of those materials that are mobile around in the plant, but it's, you know, it's absorbed where the droplet lands and taken into the cells. So it gives it a bit of energy, lasts a long time on the ground. That if it's dry, it'll rain, and the plants will be able to then stimulate root growth and pick up nutrients. So it's more or less a stopgap that provides a nutrient for a short period of time. It's not meant to be a fertilizer program that is beyond what you're putting in the ground. So let's leave it at that for now. So one of the key things is the plant makes everything out of these components. CO2 for carbon, water for a lot of reasons, oxygen, sunlight for photosynthesis, and nutrients. It then uses its genetics to biosynthesize everything. And therefore, it's always trying to get to a state of balance or homeostasis of the nutrients in the cells so that it can do the things that it needs to do with its enzymes, and its genes can produce or can stimulate the production of everything the plant needs. And everything the plant needs sort of consists of these things: vitamins, hormones, fats, proteins, amino acids, and it's the carbohydrates and sugars produced in photosynthesis that allow the plant to get carbon to build all of these molecules. As you know, I believe the plant knows what to do, and if we look after photosynthesis, the plant will do what it can, and our job is to provide nutrients where we are able to. Now, we're just at the stage where the plants are coming up. The seeds are germinating. Over the next while, we're going to be faced with abiotic problems from drought, salinity, too much sun, not enough sun. We're going to have defense mechanism against organisms. We're going to have mechanisms to deal with herbicides that are absorbed by plants and have to be metabolized. And those nutrients and that autotrophic approach will produce things like antioxidants to deal with oxygen problems, things like glutathione from sulfur, tocopherols, vitamin E, produces all kinds of enzymes. It produces compatible solutes that are used to keep the cell from performing, or keep the cell performing when it's under drought or heat stress. It forms all the hormones which influence the physiology of the plant, and it looks at homeostasis of macronutrients, or macromolecules, and ion homeostasis. In other words, the materials that are used as straight ions. And there's variability in the soil. Nitrate, sulfate, chloride, and boron move. They're anions, so they're negatively charged. They move very readily. But when it's dry, they become somewhat immobile because there's no moisture, and the plants need moisture. You know, plants drink, they don't eat. There are those that are immobile: phosphorus, copper, iron, manganese, and you'll notice manganese, zinc, and phosphorus are in build because those are immobile in the soil. They don't move a lot. Manganese, iron, copper, iron, manganese, and zinc are metals. They're not soluble, so there's very little in the soil solution at any time, and phosphorus just gets tied up by a whole bunch of other minerals, depending on the pH. At high pHs, they get tied up, and at low pHs, they get tied up by things like aluminum and iron. but mostly aluminum here on the prairies. At the high end, they get tied up by calcium. Within the plant, though, things vary. Nitrogen, phosphorus, potassium, magnesium, molybdenum, and chloride are mobile. In other words, the plant can take it from cells, old leaves and so on, move them into new leaves. They can move them from cell to cell, likely through the plasmodesmata that connect the cells, whereas other ones are immobile in the plant. Now, I should point out that when we talk about mobile and immobile in the plant, whenever the nutrient is coming up the xylem from the roots, it's mobile. But once it gets into the cells and gets incorporated into either enzymes or into the cell wall or into DNA, something like that, it is not mobile anymore unless the plant kills those cells like it does when it's senescing or when it's actually really deficient and has to kill cells in lower leaves or older leaves and move them. So all of these are mobile in the xylem and moving up, but once they get established in the cells, they're not. So it depends on your shortages and deficiencies as to which ones will be an issue and which ones are more important than others. pH. You know, when I sort of got into this area of plant physiology, I never really understood pH. Now I do. It is really, really key to the availability of all the nutrients in your soil system. Some general rules: if you got a pH between about 6.5 and 7.2, man, you've got it made in the shade. That is perfect for availability of virtually everything. As the pH goes up, it becomes more basic. Take a look at the micros: copper and zinc, boron, manganese, iron, even phosphorus up here. They become less available, except for molybdenum. Molybdenum becomes more available, and it's not really available when you get down below about 5.5. But the other micronutrients become more available as the pH goes down. But the macros like manganese, calcium, even a little bit of sulfur, not so much potassium, but phosphorus become less available, and you get solubilization with phosphorus, you know, of aluminum and iron, which ties up things. So pH is really important. And if you guys aren't taking soil tests and know what your pHs are and the variability, you're really, really missing out understanding your soil, to tell you the honest truth. And here's a rule of thumb that, you know, Elston Solberg always uses, and he's got good credibility in my mind, is that if your pH is 6 across your field, you probably got a variance of 1, which is a 10-fold difference between 6 and 5 or 6 and 7. So if your pH is varying a lot, and depending on how you take your soil test, you may have to really think about how you're going to apply your nutrients. If you're going to ban them, you're going to foliar treat them so they're not tied up because of pH. We're getting close to the end of sort of the basics here, but the other thing we have going on here is in minimum till is we have stratification, because things like phosphorus and potassium, we'll use as examples, we know are not very mobile. So you can see that once you get down below about 2 inches, it's really not that readily available, because we got no way to move it down, and we keep putting the phosphorus in the same place and the potassium in the same place and the zinc in the same place and the manganese in the same place, and it can't move down. So as a result, we can run into shortages, because as the surface dries off, the plant has no option but to go deep for water, because water drives everything. Without water, there's no transpiration. Without transpiration, there's no movement of nutrients up into the system, and there's no pressure to move nutrients and byproducts of photosynthesis in the phloem and distribute it through the plant. Water pressurizes the whole cell, the whole system, and potassium and chloride and other nutrients are important in terms of turgor pressure, or pressure on the cells which grow cells. So we have to take into consideration how those vary as it gets drier, and that's why drought is a real serious problem, because these nutrients, which are highly available in the upper surfaces, and most roots feed in the top 10 to 12 inches. When they're going down really deep, they're generally going down for water, simply because there's no oxygen there and it costs a lot to go down for water. Anyhow, let's get down to where we're at now. We got seed in the ground. We're starting to germinate. You can see here's the coleoptile starting to go. The shoots are taking off. The roots are starting to grow, and they grow very nicely. Wheat here, you can see the root hairs get going early. And before long, you'll have your coleoptile up. The tip will open up. Wheat will, well, your first true leaf will pop out. Cotyledons will pop up in your legumes and your broadleafs. And you got your roots. This is a canola root. You can see that the canola roots, even as seedlings, tiny seedlings, you can see that these lateral roots near the top grow up almost horizontally and grow for that way for life. And that's why canola can scavenge nutrients like a darn up in the upper surfaces, because the roots grow laterally where the nutrients are. If you look at wheat don't. Wheat roots grow at about a 45-degree angle. These are their the seminal roots, the early roots, and then the crown roots will grow from the crown near the soil surface, and they'll grow at a stronger angle. But canola can scavenge and has great enzymes that extracts nutrients like a darn. After a canola crop, you got to really look at your nutrient package going into the following crop. I want to give you one tip when you're out in the field and looking at plants. This is stuff I did when I was looking at the effect of fertilizers on plants, and you can see that this is urea. I use this urea because I know urea causes toxicity in plants if you give it too much. So when you're out checking your fields and you're looking at emergence and you're seeing variability of the shoots, dig down and take a look at the roots, the roots coming off the seed or later on. If they're burnt off like this, that tells you that you've had some toxicity and maybe your dosage is too much. This is going to happen probably with urea, maybe with potassium chloride, if you put too much in the seed row. If you're side banding, it's not nearly as risky in mid-row banding. By the time the roots get there, there's a lot of dilution takes place, and you're probably not ever going to see that happening. It's going to only happen where you have a really narrow opener, it's fairly dry, and you've got a lot of nutrient in that opener or seed row. Now, what are those roots doing there? Well, the roots really control what's coming into the plant. So they're putting out carboxylate, phosphatases to extract nutrients from the soil. Those enzymes and exudates knock things off the cation exchange, release them into the soil, release iron, release manganese, and are exchanging them. And then transporters take them into the plant. Here you can see iron, manganese, zinc, and there are different transporters for different nutrients. You can see that this is a different transporter. The plant is pumping hydrogen here to acidify its rhizosphere. This is all going on genetically to help with the homeostasis in the shoot, and the shoot and the rooter are communicating all the time. You can't see this. This is why I'm showing it, and you have to just believe that the plant is doing this because the plant knows what to do. So in reality, here we are, right here. This is the typical growth curve until reproduction, then we start grain filling, whether it be canola, whatever. It just varies depending on the crop. So we're right here. So we got seed germinating, we're putting our ground, we got our fertilizer into place, and we got to get this thing rolling, because once this takes off, it's got to build all its reproductive parts, it's got to build all its stems, all its leaves, all its roots. By the time it gets here, and then fill it, because if it doesn't have that done before we go into reproduction, the focus is not on producing biomass, the focus is then on filling. And anything that isn't ready will be killed off by the plant in order to make success in filling. So we got to get there. And here, for example, a lot of people don't see this. This is wheat at stage 31, which is 1 centimeter above the ground. It's about the 4-leaf stage. Let's put it this way. This is what the actual seed looks like right here at this point in time. So this is what you're growing. You want to grow this seed, and you got to produce all the carbon, all the photosynthesis, and take up all the nutrients to grow that seed up until it gets here. And then the plant will pull everything that is produced here up into here to help feed it along what it's taking into the roots. But as soon as it gets there and it pollinates, hormonally it changes, it shuts down the amount of nutrient it'll supply to the roots. So you want to have your plant built by the time you get to that stage. This is a new way of looking from a physiological perspective at nutrients. And you can see that the red ones are the ones that are in active build. And we'll just go through this quickly. Group 1 are carbon compounds. Nitrogen and sulfur make up the proteins largely along with carbon. So that's what builds along with P. And these aren't exactly exclusive. The phosphorus is used in a lot of genetic material and so on in building the cell as well. So we put some N in there because the plant is always needing a little bit of N, and in the end helps get other nutrients into the plant. Then we have energy storage and integrity, phosphorus, boron, and silicon. We put phosphorus and boron there in combination to build the cells. There are those nutrients that remain in the ionic form. They're not reduced. Both nitrogen and sulfate, nitrate and sulfate have to be reduced before the plant can use them. So it takes a lot of energy, and that's why we put phosphorus in there. ATP production is key for running everything in the plant. These are used in the plant in the ionic form. Potassium, which we have in there because potassium can get tied up when it gets dry, or if it's too wet or the roots aren't growing. And we put zinc in there because zinc is short across the prairies. It's short worldwide in many parts of the world. It's a metal, and we're taking out our metals and we're not putting them back. And that resource or reservoir will slowly get depleted over time. Sodium, we don't worry about because having too much is more of a problem. Chloride seems to be only a concern in wheat production, and I think guys that are using potassium chloride on a regular basis are probably getting all the chlorine they need, but I'm starting to see more and more in the literature that chloride's becoming a problem. These are usually a combination or enough around. Redox reactions are metals: iron, manganese, copper, nickel, molybdenum. We include molybdenum and manganese in this formulation. Copper can be a problem in real sandy conditions. Iron isn't much of a problem across the prairies except when it comes to soybeans, as far as I'm concerned. If you've got iron at high pHs and a lot of free limestone. So this is how we came about, along with our patented bioactive molecule, a formulation to support the plant in all these categories and the nutrients that are really key. And that all takes place in the cell, and the vacuole stores any excesses. So if you're putting on a foliar, it's giving the plant quite a bit of jolt, especially on those cells where it's hitting directly, say, underneath the leaves. Well, the plant, to prevent toxicity, takes it into the vacuole, which is safe, and then over time it will pump it back out and feed the plant. So that's how it deals with excesses. Now, I'm putting this in because a lot of people show it, and it just bores me spitless when people start talking about it. But I want to put it in just for a second because one of the reasons we have problems with our nutrients is that they cycle. Each one of these, nitrogen and sulfur, have an atmospheric cycle, a soil cycle, a leaching cycle, a tie-up cycle, just like phosphorus and potassium. And these cycles are influenced by moisture, by temperature, by microbial activity, the structure of the soil, and all of those are always going on. So you never know where the dickens you are in any of these cycles. So we put in some nitrogen, we put in some phosphorus, we put in some potassium in order to help the plant no matter where it is in the cycle, because the transporters will control whether it gets in or doesn't get in when we're putting on a foliar. It's not a high dosage. We don't add sulfur because everybody should be applying sulfur and looking after sulfur because almost all of our soils seem to be somewhat deficient across the landscape, and that's why we have products like Sulfur Plus to deal with that. So we're getting down to the nitty-gritty. Let's take a look at each one of these nutrients now. Well, let's take a look first at when do you use them. Herbicide timing, 3 to 5 leaf stage. I'll show you a little bit more of that. Promotes growth below and above the ground. It helps with rapid growth. It helps in metabolizing herbicides that are in the plant. It helps in terms of dealing with reactive oxygen species in unfavorable environmental conditions. And nitrogen, potassium are in easy to absorb complexes, and phosphorus in two forms. So that's sort of the timing and the logistics or the logic behind the product. About 1 liter per acre and about 50 liters of water per acre, or 20 liters per acre with ground application, and about 12 liters per acre for aerial application of water. And here are the timings for wheat, oats, canola, and so on. You can see they're 3 to 5 leaf stage, with canola 2 to 4 leaf stage, about herbicide timing V1 and 2, 4 to 5 leaf on peas, lentils, and pulses and flax. So those are the timings. Get familiar with those. Also, go to our website, Taurus website or Active Agro website, and start taking a look at the interactions. The compatibility charts are there, and this is how you apply water, your agrochemical, and your active build. Those charts on compatibility have developed, and they're on those websites. And start looking now, because you know what herbicides and what weeds you have, what herbicides you're going to use. Take a look to make sure that they're going to be compatible with active build. Spray early morning or late afternoon. The reason is you want to keep that droplet moist as long as you can for absorption. Early morning, the plant is waking up and there's a lot of nutrients starting to move, and at sundown, it's shutting down. But you don't want to go in that hot part of the day where you're going to get a lot of loss due to quick drying on the plant. Don't spray when it's hot, you know that. Avoid spraying on windy days, basic common sense. And here's one thing I really suggest you do. And if you've never done tissue testing and want to start, just do one field or a couple fields. Do a couple tests during the summer. You'll learn more about what your nutrition package is doing for your plants than you ever wanted to know, perhaps, and you'll learn a whole lot. Just try it. And if your soil tests are showing deficiencies, hopefully you're going to see your tissue analysis, and if you're treating it, you will not see it in your tissue analysis. If you're still seeing something in your tissue analysis after applying a micro as a foliar, then you probably need to do something in terms of a broadcast or a much more intensive management package, because a foliar nutrient micro package is not going to solve a problem. It will be an interim solution to help the plants, but it's not going to raise soil levels high enough unless you put on an exorbitant amount, or you put on a granular, where you focus on bringing those soil levels up. Nitrogen. Well, I don't think I have to say much about nitrogen. It makes up all your proteins, your nucleic acid, your nuclear materials, your coenzymes. The plant runs on sugars and photosynthesis. They form a backbone of molecules. Those molecules are largely proteins made from nitrogens. And proteins run the plant. Hormones, enzyme structure, everything. Biosynthesis. Oops, I already have that one there. A couple things you need to know, though. Nitrate cannot be used by the plant. So what it does, it takes the nitrate into the cell, and nitrate reductase, it reduces to nitrite, and then it reduces to ammonium, and then it moves it into the plant as glutamine or asparagine, and that's how it incorporates that. Reason I put this on is we got nitrate transporters that are bringing in the nitrate, but here, nitrate reductase. To reduce that nitrogen, we need potassium, sulfur, iron, and molybdenum. Iron and molybdenum are used in catalyzing that process. That's why we have molybdenum in there along with other things, because nitrate has to be reduced and sulfate has to be reduced. The sulfate is reduced to sulfite and then to sulfide, and then incorporate it. Both of those processes take a lot of energy. In fact, the reduction of sulfate takes more energy than the reduction of nitrate. That's why we need that ATP. And ATP is pretty well always hooked to magnesium for activation. Gives you some idea on why we have these things. Phosphorus, phosphate, nucleic acid, coenzymes, phospholipids, and ATP. Here we go. This is DNA. These are P. It's phosphorus and DNA. So the plant building genetic material, every cell has it. Every cell needs phosphorus. ATP. Every cell needs it. Phospholipid plasma membrane. All of these are phosphorus. Every plasma membrane is covered on both sides with a fatty layer in between. Just don't let your plants run out of phosphorus or that plant will not grow. You will stymie growth of everything in that plant. It's the spark plug of life. Just quickly, I did a phosphorus presentation back a couple of weeks ago. Take a look at this. When we get to lower pHs, phosphorus is fixed by iron and aluminum. They become soluble and get fixed. So that may take 10 or 15 or 20 years. That's why to be released, if ever. But when you put on limestone, you bring the P up, we can recover some of that phosphorus. At the high end, it can get tied up with calcium, not as badly, because plants can acidify the rooting zone. So if you got a pH of 8, right in the rhizosphere, the plants can pump out hydrogen, acidify that rooting zone right around the root hairs and drop the pH to help extract phosphorus when we have calcium levels there. It takes energy as usual, but it can be done. But this area right here between about, 6.5 and, about 7.2 or so is ideal for, got this wrong. This is phosphorus fixation with aluminum. This is iron. The good spot is right here between 6 and about 7.2. Then we get really good uptake of phosphorus there without much inhibition at all. And at both ends of that, we run into problems. Potash, cofactor for more than 40 enzymes. It provides cell temperature, water regulation, and neutrality of charge in the cells. Water regulation, because potassium is involved in stomata opening and closing, and turgor pressure builds the cells. When cells are created, they're small. It's turgor pressure in the root system and the shoots that expands the cells and grows them. So when we don't have potassium, we have really poor regulation of water, and we have poor growth. And it's involved in all kinds of stress management. All kinds of stress management. Just take a look at reactive oxygen species 1. Root respiration promotes phospholipids and improves membrane integrity. Strengthens cell walls, we all know that. So potassium is really, really key. And when it gets dry, potassium gets tied up in the clay lattices and becomes very difficult to extract because there's no solution and the roots are not growing and it can't get it very easily. So a lot of times we'll see, especially later in the growing season when it's really drying out, we'll see deficiencies in tissue tests of potassium later in the season, even though soil tests may show there's a lot of potassium available, but the plants can't get it. So that's what tissue testing will help you determine. And now we got in Alberta, we got a lab that does sap testing, which I think has some real potential to help us understand how nutrients are moving in the plant. Boron. There are a lot of talk about everything boron does, and it's important, but about 90% of all the boron in a plant is tied up in cell wall structure. It cross-links, and I'll show you a little bit. It's important in cell wall structure, and it's important, therefore, in everything that is growing, because every cell has a cell wall. So it's involved in meristem and root growth. It's important for rhizobium inoculation because when the plant is growing the nodule, remember the plant grows the nodule, it has to restructure all that cell wall. It takes a tremendous amount of calcium, a tremendous amount of boron to build all of those nodules. So it's really key in legumes. And it's important for defense in response to soil microbes, because if you've got a strong cell wall, then organisms can't penetrate them and get a hold. The hyphae can't get into them. And last year I did a whole pile of trials by incorporating boron into sulfur plus to see if we could control Aphanomyces and clubroot by strengthening the soil wall. Well, unfortunately, those trials for us didn't work out, and we didn't get any protection against those two organisms, even though laboratory studies that by students at the university showed that we got some relief. So it's really important for some plants and some protection against certain soil microbes. And you can see here the boron is connected here between various tissue in the cell wall to provide a cross link, especially with pectins, and that's why boron is generally more important in broadleaf plant cell growth than in monocots, because there's much more pectin in broadleaf plants. And it's this material here that I can never pronounce that is really key in the cell wall, and that's where most of the boron is tied up. So that's really the key component of boron. Here in the roots, as I talked about, deficiency causes leakage of potassium, as well as allows the plant to build the actual root. And here are the nodules. You can see that these nodules are produced by the plant, and then they feed the nitrogen back down. And one of the things that I borrowed this from Greg Patterson at A&L, he's finding that boron is really, really critical for good soil microbial activity. And I think this is an emerging area that we don't fully understand yet, and how these nutrients play a role, and how mycorrhizae play a role in providing things like the zincs and the borons, and coppers that are difficult to get at distances from the root. The mycorrhizae can go out and scavenge much further than the root here. And as we learn more about the interactions, and we look at other mycorrhizae organisms, fungi, that can be beneficial to plants, I think we'll see that they need these nutrients as much as the crop does, or maybe more, and that the whole balance between the soil and the plant will be better understood. But right now it's pretty well in its infancy as far as I'm concerned. Just about to the end. Manganese. We put this in here for a couple of reasons. Number one, it's involved in the first stage of photosynthesis, and that is the production of oxygen, and I'll show you a little table. It's involved in managing reactive oxygen species, or oxidative stress, or what we in humans would be called free radicals. And it's involved in a whole bunch of enzymes. And for soybean growers, mostly I guess in Manitoba, manganese is really key in soybeans because of the way soybean manages its nitrogen. So if you've got deficiencies in Manitoba in manganese, which I don't believe there are significant ones, or I'm not aware of them anyway, then consider testing some manganese and put on some strip trials or something. But let's take a look. Here's photosynthesis and photosystem 2. You can see right here, the first thing that has to happen is water has to be broken down. And there's a lot of reasons that hydrogen is used to then produce ATP and so on. But what is involved in that is this manganese, lots of manganese and calcium. And there should be some chloride in here somewhere as well. It is this oxygen evolving complex that starts the whole process off. So when we are a young plant, and if manganese becomes a problem because there are 3 states of valence states, and the plant only uses in the +2 stage, so there can be deficiencies just based on environmental conditions, and manganese becomes tough for plants to get at higher calcareous, under higher calcareous soil conditions. So that goes into the product. And here's Something I always talk about in terms of stress management, these things called superoxide dismutases, they take these free radicals and they convert them to hydrogen peroxide and then to water. And then the hydrogen peroxide is then catalyzed by other things. But manganese sod is in the mitochondria in the chloroplast, iron sods are in the chloroplast, and copper zinc sods are in the chloroplast and the cytosol. And now they believe they found a nickel sod. So these are involved in scavenging all these free radicals that occur in the plant along with a bunch of sulfur compounds. They're really key. It's now starting to be talked about a lot more than it used to be. I've been pumping this for a number of years, and you can see that zinc is involved with copper in this particular copper zinc one, but manganese and iron have their own sods. Zinc in all 6 enzyme groups in the plants. It's important for germination and enzyme activity and reactive oxygen species scavenging with copper. I showed you that combination. There it is again. And here's the situation with zinc in the prairies. You can see that this big chunk here, largely deficient in zinc. Alberta has zinc deficiency. Maybe it's worse than we think. These were done in 2014. Manitoba, in some of the stuff that I've seen recently, showed about 61% of soil samples from IPNI were deficient. About same in Saskatchewan, and only about 31% in Alberta were showing deficiency of zinc. But man, our wheat studies this year showed that we maybe have more than we thought. Something to consider on your farm. Do some tissue tests. Zinc pulls up real easy, and looking at tissues and soil tests. And zinc can be affected by climatic conditions, interruption or interaction with other nutrients. Soil composition can influence this. Carbonates tie it up. Higher pHs and microbial activity can have an effect in terms of releasing it from organic matter. The same kinds of things that affect availability of almost all of our metal nutrients. And here are just some sensitivities, insensitive to them, sensitive, or these crops more so. These are mildly sensitive. Wheat seems to be, barley, alfalfa. So things like canola, likely it's not in here, but it looks like some of the mustards, for example, which are crucifers. Peas use a lot of zinc, but it seems to be able to get it, and rye, of course, can extract it. And that may be why rye does quite well in some parts of the prairies. I'm not sure. Deficiency are affected by cold springs, fine texture soil, and calcium carbonate and elevated pH, which we've talked about. Now, when we talk about phosphorus and zinc together, we have an issue. The two have a relationship. If you have too much phosphorus, it ties up the availability of zinc. If you've got too much zinc, it may tie up the availability of phosphorus. So you have to look at that balance. If you're putting down a lot of manure, for example, or extremely high phosphorus levels where you have zinc deficiency already, then you may have to pump up your zinc levels in order to provide the plant with enough, because it's in competition with that phosphorus. Talk to your agronomist about that particular aspect of managing the zinc and phosphorus interaction. Lastly, molybdenum. Very small amount, but it only takes a tiny bit. 25 grams per acre will solve a moly problem in acid soils. It's only in 5, maybe 6 enzymes in a plant. Nitrogen management, nitrate reduction, I talked about that. But the other important thing for legume growers is that when the plant fixes nitrogen, it has to break down N2, which is triple bonded. And to do that in the nodule, it has to have molybdenum as a catalyst. So molybdenum nodules are a sink for molybdenum in legumes and in acid soils where molybdenum becomes difficult to get, or under stressful conditions. You want to consider maybe more moly than what's in active build. And you can put on a straight moly application there if you're really, really deficient in your acid soils. So consider that. And it's also involved in sulfite management Now, sulfate, when sulfur compounds are recycled in plants, sulfate has to be converted or it becomes toxic. And one of the enzymes involved in that is molybdenum. It's also involved in abscisic acid formation. So it has a lot of important uses, but generally we haven't had a problem except where the pHs are low. I'll just show you this again. Nitrate reductase here is where we have the moly. In the nodules, this is where the nitrogen fixation takes place, and there's a buildup of molybdenum in here with sulfur and iron all together in a molecule, because moly doesn't ever work by itself. And this just goes to show you how a nodule actually works. And you can see we have transporters for molybdenum, for sulfate, for iron, for magnesium. A lot of people aren't aware of that and maybe don't even believe it, but the plant controls those into the nodule itself, and then it actually breaks down the nitrogen, forms ammonium right here ammonium, sort of ammonia first ammonium, and then it ships it off to the shoots. One thing you need to know is that moly has to have copper in order to form all of these enzymes that we just talked about: sulfate oxidase, nitrate reductase, aldehyde oxidase, that are involved, aldehyde oxidase involved in ABA production. And lastly, just to show you, here's the molecule molybdenum with iron, sulfur, and some cysteine. And this is what happens here in breaking it down with the nitrogen enzyme and the molybdenum cofactor, and it takes a whole lot of ATP. So that's why we put phosphorus in, we put moly in, we put some nitrogen in, we put some potassium in to try and give this plant and other things with some bioactive molecules to try and support that plant when it's in a growing spurt and there's a chance that there's a deficiency or a stress that needs to be handled and the plant can't get enough through the soil. I hope that helps. Look forward to the next one we do. Thanks a lot. Have a good spring, and we're going to have a great crop this year. Take care.